Hello, and welcome to this Edison TV interview where we're speaking with Carlos Buesa, CEO of Oryzon Genomics. Welcome, Carlos.
Thank you. Good morning, Aaron.
We're going to focus today on Iadademstat, the company's lead asset in hematological diseases. So, Carlos, to jump right in, could we start with a summary of the mechanism of action of Iada?
Certainly. Iadademstat, Iada as a nickname is easier, is a potent and selective LSD1 inhibitor for oncology and hematology. Well, the function of LSD1 is it's an epigenetic enzyme, which is basically a master gene in controlling gene expression. But it's also key to maintain the leukemic stem cells alive in an undifferentiated and proliferative state, the cancer cells. So when we inhibit LSD1, Iada basically promotes the differentiation of malignant myeloid cells to more differentiated status, which can enter into apoptosis. So it's a pro-apoptotic drug in that sense, but also is acting as a synthetic lethality agent in leukemic stem cells. So we also know that enhance synergistically sensitivity to standard AML therapies and enhance enormously the immune response. So this is basically the mechanism of action of Iadademstat. But I would like to add that beyond that, one thing which is particularly attractive is that Iadademstat, because of this mechanism of action, is mutation agnostic and is not restricted to a single molecular subtype. This clearly differentiates Iada from more mutation-specific approaches, like menin inhibitors, for instance, or other like FLT3 inhibitors, which are primarily targeting defined genetic AML subgroups.
Excellent. So we've set the scene just there. Could we move on now to an outline of the current clinical programs in AML, and particularly frontline AML, and the encouraging interim data due to be presented at the EHA conference this June?
Yeah, we have a dense pipeline in hematology, in second line, in MDS, in myeloproliferative neoplasms, but certainly the key driver program right now is in first line newly diagnosed AML patients in combination with Veneza. These programs that we call ALICE-2, to differentiate from ALICE-1 that we did in the past, where we treat the same population with only ASA, is basically providing really highly encouraging data. We have reported at EHA as the cutoff data of the abstract presentation, 100% overall response rate and 93% composite complete remissions. So that also with a very high level of transitions to human stem cell transplantation. So we are presenting in 4 weeks from now in Stockholm at EHA data which will include more patients, longer follow-up than in the abstract, obviously. So we are really excited. We expect that the data is going to be at least as compelling as the one that we have at Advance, and hopefully more. And this is something which is important because at EHA we are going to present data of approximately 75 to 80% of the whole plan, at least to recruitment, which is making this data, we think, a meaningful interim readout rather than an early signal from only a few patients. So we see that this combination that we are proposing right now is very competitive versus other emerging triplets like menin inhibitors or antibody-based approaches, because it basically has a broader applicability in terms of the, as I said, is basically working in virtually any AML patient, a stronger response rate so far, and a more favorable tolerability profile. So we are really excited about this program ALICE-2 that, as I said, is the key value driver for the program right now.
Fantastic. So taking a step back now, could you summarize the key unmet needs in frontline AML and then how Iadademstat is positioned to address these?
Well, I mean, I think that we need to remember, all of us, that 1/3 of the patients that are currently treated with the current standard of care are not responding, are refractory to this. So 1/3 of the patients cannot benefit already from the current armamentarium. On top of that, we are proposing, well, we are basically showing here 100% of responses. So this third of patients which are unfortunately not responding to the current standard of care are responding to our triplet. But on top of that, those cohorts of subpopulations of AML patients which are really responding very poorly or at all to the current standard of care, like p53 mutant patients, like NRAS, KRAS mutant patients, like myelomonocytic leukemias. All these patients are responding very well to our drug, as we saw in an earlier trial, Alice 1, where we saw that we were able to double the median overall survival of TP53 mutated patients, and we were having 100% of responses in NRAS, KRAS patients. So this creates a really strong clinical proposition. IADA could become a broadly applicable AML triplet with a particular value in high-risk patients, like the ones I mentioned, the high-risk P53, and also the intermediate risk NRAS, KRAS patients, where regulatory and commercially opportunity are especially compelling. We have done a market survey, a market access survey with KOLs and payers in the US, and we have seen that they anticipate a very good receptivity on this positioning, and because of the extremely high unmet medical need, particularly in P53, a good flexibility for a high price and peak sales that will go north of $1 billion only in the US.
Excellent. So looking beyond AML now, could you outline for us the broader opportunity for the drug candidates, for example, in sickle cell disease?
Sure. But before we go to that, which is also a very compelling and fascinating proposition, let me insist that thanks to the creator agreement that we have with the National Cancer Institute and with the agreements that we have with several prestigious universities in the US, we are also exploring myelodysplastic syndrome. We are also exploring myeloproliferative neoplasms. We are also exploring small cell lung cancer, where there are currently two clinical trials going on. One which is being led by the Memorial Sloan Kettering Center. The second is being led by the Yale University. So, I mean, really, IADA has been exploring a very broad landscape in oncologic indications. On top of that, we believe that there is a very strong opportunity in sickle cell disease. Sickle cell disease is well known. Is caused by a mutation on the hemoglobin gene. And we know, because there are beautiful examples of human genetics in Saudi Arabia, we know that there is a population which are carrier for this mutation, yet they don't develop the disease because there are other mutations which are suppressing the phenotype of the primary mutation on the beta hemoglobin gene. And this is basically producing an overexpression of fetal hemoglobin. So we know, we have data with our compounds that in rodents, in monkeys, we are able to overexpress the fetal hemoglobin, and we are also starting right now a clinical trial in sickle cell disease here in Europe. So we think that over the next quarters, we might have an idea on how this program could be also being advanced forward.
So to wrap up, what are the key expected milestones and next steps for Iadademstat throughout 2026 and into 27?
Yeah, as I say, the key driver programs is Alice 2 in first line AML. So very easy for the investors, very easy for the community. They have to stay tuned on the data that we are going to present in 4 weeks, to stay tuned on the data that we are going to present at ASH, where we expect to present virtually the final results of this study. Following that, we are expecting to go to the FDA to propose a seamless phase 2/3 program, our already named Alice 3, which we want to focus on these specific populations, P53 mutant patients, and NRAS, KRAS, myelomonocytic leukemias. This is a design that have been followed by others, by many inhibitor developers, for instance. So we are not inventing the wheel here. We expect that the regulators are going to note on this proposition. And the company goal would be to initiate this study, at least 3, in the first half of 2027. Given the unmet medical need, particularly in P53, we anticipate that we could complete the recruitment in only 24 months. And if the data of ALICE 3 remain as strong as the data that we have been producing so far, we think that that could support a potentially accelerated approval strategy. So that would basically transform Oryzon from a late clinical development company to a company with a tangible path forward towards registration and commercialization. So I think that, the next months can be transformational for Oryzon, and I think that these are the more basically valuable inflection points that we will expect.
Looks like an exciting few years ahead for Oryzon. That brings us to a close for today, but if our audience would like to learn more, we direct them to the Edison website where our research is freely accessible. Carlos, thanks for the insightful discussion today.
Thank you very much, Aaron. It has been a pleasure as always.
Likewise.